Yeah, thank you very much for the invitation. It's really a pleasure to be here. And I hope it's not a limitation to be an MD, actually, with all this physics and all that other stuff. So it is quite a challenge, I think, to step from viral entry into ultrasound in epileptic rats. And now I will challenge you with another topic, that's chlamydial infection. And I will just give you, with 2 slides in the beginning, a brief overview why it might be a good start to image first and then try to get more into the details by looking at the metabolic composition, let's say, between infection and host in intracellular chlamydial infections. And as you all know, probably not all microbiologists or cell biologists, I will just give you a very short introduction into the life cycle of chlamydia, because I will show you images which always reflect these typical pictures, and you do have to decipher between an extracellular form, that's the elementary body, which goes into a human host cell, mainly an epithelial cell. There forms this typical inclusion, which is almost as large as the nucleus, where the reticulate bodies evolve, and these are supposed to be the really metabolic active phases of the pathogens. And if you let them just sit into the cells for 2 or 3 days, then this cell is filled up, where these reticulate bodies, they redifferentiate to these infectious elementary bodies, and then these cells get lysed, and you have a new cycle of infection. In the clinical fate, you do not always know whether this is really the cause which occurs, or whether you have other confounders which would also result in a persistent infection, namely that you have a host immune response, interferon related, which might drive the infection into a so-called persistent infection, where you have a large inclusion, you have these reticulate bodies, but there's no metabolic activity anymore. You can also induce that if you have sub-inhibitory concentrations of antibiotics, or even if you treat them with penicillin, you also can drive them to persistence. And the metabolic background of all that is not known, and the difficulties is quite obvious. How can you decipher between the cellular metabolism and the chlamydial metabolism, because it's directly linked together? And that was the reason why we thought about doing imaging techniques first, namely 2-photon microscopy, to try to get a little bit more insight what the metabolics is coming from. As a medical doctor, I also have to show what the consequences are. If you have an undiagnosed chlamydial infection, at least in females, it's the most severe form afterwards that you can have infectious infertility. And it's supposed to be that, and at least in Germany, almost 100,000 females suffer from that. Of course, there are other causes of infertility, but this is preventable, and therefore it's really important to know the background of it. What I want to show you here is, I'll come back to that in a minute, that normally our models which we have is the normal incubator trying to cultivate these bacteria and the cells with 37 degrees in a normal oxygen atmosphere. And if you look into this surgical image, you already see that normal oxygen will not be the environment where you normally will find these pathogens. And this is just a small draft. That's the idea behind. You have normally a lower genital tract infection which can ascend and then result in inflammation into the tubes. And you have several confining factors like the hormones, like the microbiome, which is a huge topic at the moment, of course, but also the oxygen availability. And if I talked to a physiologist years ago, and I thought about what is the normal environment which you would find, and he said it's something between 1% and 4% oxygen, so it's not that what we already applied all over the time in our lab. So the experimental setup which we had is very simple. We just grow the pathogens in a normal cell culture, also in HeLa cells, and what you normally observe are these large inclusions. So this is an electron microscopy where you see a lot of these already redifferentiated small dots. These are the infectious elementary bodies. And if you apply interferon gamma, you can hardly see the inclusions anymore. So there must be something going on which prevents, of course, the growth of the bacteria. If you do exactly the same experiment, not under 20% oxygen, but under 2% oxygen, you see that the inclusions are even larger. And if you want to inhibit the growth of the pathogen applying interferon gamma, it doesn't work. And this was really astonishing for us, because if you have in mind what the pathology might be, that you have to clear the infection very early, but you do that in an environment which is oxygen depleted, then you will hardly come across that. And we titrate that. If you have 5% oxygen, you can control the infection, but if you go lower, and this is a really sharp cut in 3%, even if you give high dose of interferon gamma, you cannot restrict chlamydial growth intracellularly. This is just the background, but we thought this must have something to do with metabolism, that in those cells, of course, bacteria have not the capability to grow as large as they would have normally in a normoxic atmosphere, or if you even make them hypoxic. So we go through that, and as most of my previous talkers already said, it mostly starts with a custom-made machine, and this is a custom-made hypoxia, but also randomized, let's say, lab, where we are with guys from the physics, but also from the anatomy, and we designed such a 2-photon microscope. And just briefly around the background, we just looked, and this is a huge limitation to all of to those of you who look at particular interactions between antigens and antibodies. We just look on the autofluorescence of cells and also inside the inclusions, because we claimed, and this was done in tumor cells before, that based on this autofluorescence, which you find in NADH or NADPH, you can distinguish at least metabolic active sites in the cell from not metabolic active sites. And the advantage of the 2-photon microscopy is that you can do that in living cells, yet you have less phototoxicity, and you can look at least for 200 micrometer into the tissue. If you go into the biochemistry, of course, this gets a little bit more complex. And this is just to show you that if you look for the reduced forms of NADH, you claim that you can have either to do with mitochondrial activity, energy metabolism, but of course you are also on the side of the redox system, and this is a huge limitation if you look in autofluorescence of 2-photon microscopy. And it's not completely clear whether that is the truth that you just look at metabolism, and therefore it's completely necessary that you have a second technique, or even better, several techniques afterwards, where you can validate your imaging findings to really say that what I saw at the beginning has something to do with metabolism. And one of the measures you can do is to determine the fluorescence lifetime, and this is on the basis that if you look at the autofluorescence, you get the fluorescence decay curve. And based on the interactions, the protein binding of the NADH or NADPH, you have longer or shorter lifetimes, which you then can measure. And just to give you an example how that works, at least in chlamydia infected cells, this was done very simply. At the beginning, we took non-infected cells and in infected cells just to see whether we can distinguish the different compartments in the cell. And this was just done by looking at the photon counts of the autofluorescence. And what you can nicely see is that you can distinguish different photocounts for the nucleus, for the chlamydial inclusion, and for the mitochondria. And this is very simple, but this shows us at least that we have a kind of spatial resolution just based on the autofluorescence. And this you can, of course, then try to do in a more statistical way afterwards. And this is the T2 value, which reflects the fluorescence lifetime of NADPH. And as you can see here, at least the chlamydial inclusion has the longest T2 value compared to the cytosol or the mitochondria. And this allows us already in these infected cells to define a specific compartment, which is the intracellular bug. Looking now into the temporal resolution, of course, we were interested to see whether we can have any kind of kinetics which reflect the intracellular growth of chlamydia. And this is also very simple. You look here on the growth of chlamydia just on the area which you can measure inside, so the size of the inclusion. And as you can observe, after 24 to 36 hours, you have a peak of the inclusion size. But if you look on the T2 value, so on the metabolic activity of the cell by the autofluorescence in the 2-photon microscopy, you can see already 12 hours earlier by measuring that you get a peak. And this probably reflects the metabolic activity inside the inclusion. So you see here it's measured inside. So you have a shift from smaller fluorescence lifetimes to longer fluorescence lifetimes, indicating that by the interference with the NADPH with the different proteins, you get a higher metabolic activity inside the inclusion. And now, as I said before, it's so important to really validate that with your functional systems, to really see that it has something to do with metabolism. It's not either an indicator maybe for other mechanisms which are underlying by this autofluorescence. And therefore, we applied different conditions. So we had the control, where we saw a nice increase in the fluorescence lifetime. And then we applied those where we restricted the glucose concentration, or we blocked the electron chain. And you have already smaller inclusions, which you can see here. So they markedly drop. You have also reduced recovery, meaning that the bugs are growing less worse. And this is completely reflected also fluorescence lifetime inside the inclusion, that you see those, in those inclusions where you measure then here, the T2 value, they are significantly decreased. So what does it mean for the persistence infection? It might, for some of you, just be what you hypothesized before, that you can say you can now measure it, and exactly what you can do if you look for the normal infected cells and you apply the interferon gamma to it. And this is just another way to build the ratio of free to protein bound NADPH. But in here you see that you also have a decrease in the fluorescence lifetime, and this is completely reflected by the inclusion size. So that means by just measuring inside the inclusion the fluorescence lifetime of the autofluorescence NADPH, you do see functional differences between actively growing and persistent chlamydia. And as I said before, of course, you have to also validate that and look whether you can see that in another functional assay. And this is, as I also mentioned, a limitation in chlamydia, because it would be best to now bring out the chlamydia and just measure what is host cell and what is the bug. This is not so easy to do. So what we did, or we tried at least at the moment, to look in the seahorse bioanalyzer to measure the whole metabolic activity within the cell. And this is the extracellular acidification rate, which you can do so by measuring non-infected cells. You can see the glucose metabolism and the glycolytic capacity, and then afterwards you just block the glucose uptake, and then drops down to the non-glycolytic state. And if you infect the cells and you get these large inclusions, the whole metabolism of the system, so host cell and bacteria, increase dramatically. So you have enhanced glucose metabolism, enhanced glycolytic activity. If you bring them into persistence and you have these small dots here, then it's almost down to zero. That means that the whole system drives the metabolism itself. That means bacteria induce the glucose uptake and the glycolytic capacity. And if you drive them into persistence, the cells are metabolically more or less downregulated, and you don't have these large inclusions anymore. So looking at a more physiological point, and this is that you see enhanced growth of chlamydia under hypoxia. This has been shown some years ago by colleagues in Denmark and ourselves, and this was surprising because chlamydia has been grown for the last 20 years in a lot of labs in the US and also in Europe, but all people just applied the normal concentration of 20% in the incubator because it was normal to take the normal incubator. If you look where you normally find this bacteria, then it's a hypoxic environment. And if you look what cells you use normally cancer cells, HeLa or Hep cells, and they are also driven to hypoxia. So that means that they are even prone to grow under hypoxic conditions even better than under normoxia. And that's what we saw, is that if you take the same, it's as simple as I said, take the cells, put it in a 4% oxygen chamber, and you dramatically increase the growth of chlamydia. But it's completely unknown what the reasons for that is. But if you look in the literature, you saw that also for Coxiella, that they grow even better extracellularly if you put them in a hypoxic environment. You know that for viruses that you can enhance their replication by just putting the cells into hypoxia. And you saw that also some immune escape mechanisms are even favored if you have that in a hypoxic environment. So obviously it's a physiological thing how bacteria adapted to the physiology, let's say, or to the environment where they survived best. And this has been neglected a lot of years, but in the last 5 to 6 years, there's a lot of literature coming out looking specifically at that part. So looking now what comes from the host and what comes from the pathogen, we applied again the two-photon microscopy, and we looked either, because if you have an infected cell, you have to look in the nucleus because you can't see any cytoplasm anymore. And what you can see, of course, is that if you have a hypoxic environment and you look into the intensity of NADPH, this increases under hypoxia for itself, but you have no further increase here if you look in the host site by an infection of chlamydia. If you look in the inclusion itself, you can see that this dramatically increases the intensity of the NADPH, and this indicates at least that most of the metabolism occurring in an infected cell, not inside the host itself, but in the large intracellular chlamydial inclusion. One system that has been intensively studied in cancer cells are the mitochondria, and we were just wondering whether this, as a potential factor for the metabolism, but also a very important compartment for balancing the redox system, is impaired under hypoxia. And what we saw is, of course, what is already known, that if you look by very unspecific, I would say, TMR staining, this is a limitation, that under hypoxia you get an increase in this mitochondrial membrane potential if you don't make too severe hypoxia, because then the mitochondrial membrane potential is decreasing, and we don't have any effect due to the bug which we apply. But interestingly, if you have at the same hand looking on the ROS signal, and this has been nicely shown a few years ago by Agathe from the Institut Pasteur, that you need a little bit of ROS for enhanced growth of chlamydia, but due to the hypoxic state where you have a lot of ROS already in the non-infected cells, somehow this seems to be downregulated in the infected ones, meaning that you need a certain level of ROS signaling in a cell which is best for chlamydial growth, but if you would have too much, which is apparently the case in hypoxia, it seems more beneficial for the bug to downregulate that. And the question was, how is the bug doing that? And we are not completely sure whether the bug is doing that or the host cell is doing that, but if you look on the ROS signal, you have this decrease in ROS signaling in hypoxic cells. And what we now integrated from data which we had from mass spectrometry, that we saw that while normally in hypoxic cells you don't find any glutathione, which is a nice contradictory molecule to really balance enhanced ROS signal, we do see a significant upregulation of glutathione in these hypoxic chlamydia-infected cells, meaning that apparently the system is balancing somehow for the enhanced ROS generation under hypoxic conditions. And as I said, the imaging was or is the starting point, at least in our system, and it has to be the starting point because we now have to go into the decipheration between is it the bug or is it the host cell. And this is just to show you how we move on with that we look now on the transcriptional regulation of the pathogen and doing this by transcriptomic analysis. And I don't want to go through the details, but what we see apparently is that also here, energy metabolism, but also the redox system are the most important hits which we find, meaning that under those conditions where we have hypoxic environment, the bug is adapting by regulating their own energy metabolism, but also the redox system. And we're now going into detail how this works in the concert with the host response. And what does the host really contribute to the enhanced growth under hypoxia? You all know, or you might know, the hypoxia-inducible factor is really the driving force to drive either in cancer cells or also in inflammatory conditions the homeostasis of the cell. And this is not unexpectedly, but has been not so intensively studied so far, that you don't really have an effect of the pathogen, but you have a general mechanism for enhanced growth under hypoxia by upregulation of GLUT1, LDHA, and PDK1. And this means that the environment is improved for a lot of pathogens because the cell is just taking up more glucose and is driving the whole metabolism under hypoxic conditions. And this is just shortly summarized here. If you look for intracellular growth of bacteria inside a cell, you do have much more uptake of glucose. You do have, on the other hand, a shutdown of the mitochondria, which means that you have somehow no contribution to the TCA cycle anymore. And at least for chlamydia, this seems to be favorable, that we have a lot of energy going into the inclusion and is not really anymore involved in the cell energy metabolism itself. And to bring that to the end and integrate also the mass spectrometry data, you can then end up from this very simple two-photon scanning at the beginning to a very complex picture where we now integrate the data which we have from the seahorse analyzer, from the mass spectrometry, from the transcriptomic analysis, and then we end up seeing that this redox homeostasis, which is regulated apparently mainly from the host cell, but also the HIF-1 alpha response, which is mainly driving the glucose uptake and also the abrogation of the lactate, that those is the driving force in chlamydial intracellular growth. Okay, I would end up saying that the two-phone laser scanning microscope is a useful tool to predict the metabolic activity, although its limitations in the autofluorescence, which is not so highly specific, but we can do a clear separation between host and pathogen-related metabolic activities. And at least for chlamydia, it's a clear indication that in hypoxic conditions, the pentose phosphate pathway and the redox system is implied. And I think one of the major steps is, and this has been not been clear today, what the mitochondrial function really contributes to intracellular growth of pathogens. So whether the deprivation, the decrease of the membrane potential, might be beneficial for chlamydia for intracellular growth. But to be honest, I mean, imaging is one side of chlamydial biology, but one of the most important sides. And if you are a bacterial guy and work on E. coli or on other bugs, it's not so fancy maybe, but for us it's really the most thrilling stuff at the moment to get these bugs so genetically modified that we can really knock in or knock out something intracellularly, that we can look specifically on certain pathways and can distinguish between those factors coming from the host cells and decide whether this is important for the growth of chlamydia or not. Finally, I would like to thank all the people involved. Inga did the transcriptome analysis, Nate did all the 2-photon scanning microscopy, Ken is involved in the whole genetic stuff. And of course, I would also like to thank all the funders which made it possible to work on both sides, on the hypoxia and immune regulation side, but also on chlamydia research. And I would like to thank you for your attention.